Today I will be talking about gene promoter methylation in advanced tumors. And so the cohort what I'm looking at right now consists of 378 advanced or metastatic cancers. And these cancer samples are part of a research program called Personalized OncoGenomics program. And this is at the BC Cancer in Vancouver. And this research program aims to do whole genome and transcriptomic sequencing of metastatic tumors to guide clinical decision making. So there are over 1,000 tumor samples which have short-read sequencing, and a subset of these samples also have long-read sequencing, and this is the nanopore cohort, how I call it. And as you can see on your left bottom, that the most frequent tumor type in this cohort is breast cancer and GI cancer, but we also have other different tumor types, even rare tumors in this cohort. So we are particularly interested in allele-specific differential methylation. So by doing phasing, we can actually look at differential methylation on the different alleles. So in normal tissue, there are some genes where the promoter is active, so there is no methylation on any of the alleles for this gene promoter. But in tumor, we often see that one of the alleles becomes hypermethylated, while the other allele is lost, usually due to allo-age. And I'm going to call this hemizygous aberrant methylation, or HAM, in my presentation. And it is also possible in the tumor that only one of the alleles is hypermethylated, while the other allele remains hypomethylated. So this can result in haploinsufficiency. And there are also promoters which are inactive in normal tissues. So both of the alleles are hypermethylated, and the tumor we see that one of the allele becomes hypomethylated while the other allele is lost. So that leads to gene activation. And we also can see that one of the allele remains hypermethylated while the other allele is hypomethylated. So when we started to look at this, we wanted to look at the differential methylation across the whole genome in these cancer genomes. And what we found that over 4,000 genes show allele-specific methylation, either ADM or HAM, in at least one POC sample. And the majority of these samples actually have genes which show both ADM and HAM, and over 1,000 which show only HAM. And these are the genes which are usually in allo-age regions. On the top plot, what I show you is the genes which most frequently show ADM. And at the bottom plot, you can see the genes which frequently show HAM. And I also colored the genes based on the OncoKB annotation. So the blue one are the tumor suppressor, the green one are the cancer-related genes, and the red one is the oncogene. So as you can see, many of the genes which show differential methylation in the tumor samples are not associated with tumorigenesis. So it will be interesting to see how these genes contribute to cancer development or treatment resistance. When we look at the chromosomal localization of the genes with differential methylation, what we found that they are distributed across the whole genome. Although we do see a few clusters, for example, if you look at the chromosome 18, 18q, there are several genes which show frequent HAM in more than 10 samples, and these regions are known to be often have allo-age in the tumor samples because these regions contain tumor suppressor genes. And also there are a few 6p regions which have several genes which show ADM in more than 10 samples, and this is also described as a region with aberrant methylation. So we found that 56 tumor suppressor genes show differential methylation in these tumor samples. And the most frequently, the gene which shows most frequent differential methylation is the APC gene, which is a known tumor suppressor and then is known to involve in the Wnt signaling. And this gene shows both ADM and HAM as well. But there are other genes which are involved in Wnt signaling, for example, SFRP1, SFRP2, which also show differential methylation. You can see them at the bottom plot. And there are transcription factors, which MGA, GARA2, SOX17, which also show frequent differential methylation. And at the bottom, you can also see that there are two genes which are known to be involved in DNA repair, more specifically homologous recombination repair. These are BRCA1 and RAD51C. You might be heard about these genes. And these genes are important to look at, especially in the tumors, because there are some tumors which are deficient in homologous recombination deficiency, and we know that this deficiency, HRD, is often the result of a mutation in BRCA1, BRCA2, or other HR gene. But it is also important to look at these tumors because they are sensitive to platinum and PARP inhibitor treatment. So we can actually offer some treatment for these patients where if we can identify that the tumor is HRD. But we also often see that there is no causative mutation of HRD, so there is no mutation in HR genes. So we could show in a smaller cohort, what we published in 2024 in Cell Genomics, that these tumors which show HRD phenotype often have BRCA1 or RAD51C promoter methylation. And so in the top plots, you can see the promoters of these genes and the methylation in this promoter region. And what we see is that the methylation is usually somatic because, so I'm showing the tumor alleles in purple and the blood alleles in the bluish green. And as you can see, there is a methylated allele and the other allele is lost due to LOH. And these samples have high HRD tech score, which is a measure of HRD, and also show a low gene expression compared to the rest of the cohort. And we were interested in looking at the clinical response for the patients which have BRCA1 or RAD51C promoter methylation. And so here I'm showing the treatment duration before sequencing, which is the pre-POG, and after sequencing, which is the post-POG. And the platinum treatment is in gray, and the PARP inhibitor treatment is in purple. So what you can see for these samples, for these patients who have BRCA1 or RAD51C promoter methylation, they have a long platinum duration and good clinical response, usually partial response or stable disease. And the stable disease for metastatic patients is a very good response. So we generally see that the patients who have promoter methylation, they tend to respond to platinum treatment, especially with RAD51C promoter methylation. The interesting thing, what we also found in 3 breast samples which have a hypermethylated gene promoter, that there are indels in the promoter region, and we found two samples with indels in BRCA1 promoter and 1 sample with RAD51C promoter. And these indels are different in their lengths, but what is common is that they are all subclonal and trans with the methylation. So the indels are present on the methylated non-methylated reads. So that's why we suspect that the cells with these indels might be emerging subclones which are resistant to treatment, and this is a mechanism for a reversion of methylation. And there was another mechanism what we found potentially for HRD tumors, and that is the hypomethylation 5' to the RAD51C promoter. So on the top, I'm showing 5 samples, 5 patients, but they are all HRD, but the cause of the HRD is different. And in the first two patients, there is no RAD51C promoter methylation, and as you can see, 5', the CpG sites are methylated. And the bottom one is a patient with RAD51C promoter methylation. And the last two patients have an interesting thing that the CpG sites 5' to the promoter are hypomethylated, and the last patients have hypomethylation in this region, but also RAD51C promoter methylation. And what we think is happening here, that the hypomethylation in this region might lead to newly accessible transcription factor binding site and alternative isoform expression. And we look at the short-read and long-read RNA sequencing, and we could tell that the two patients, which in the top two in the bar plot, they have high expression of a alternative RAD51C isoform, which shown at the bottom corner, the second isoform, which have only exon 1 and exon 2, so this might be a non-functional RAD51C, which leads to HRD. So in summary, by phasing, we could look at allele-specific differential methylation in gene promoters and found many genes which are currently not associated with tumorigenesis. And we could also see that many samples with HRD have BRCA1 and RAD51C promoter methylation, and these patients might respond to platinum treatment. But there are also indels, there can be emerging indels in the promoter region, which might be resistant to platinum treatment. And also there is a new mechanism for potential HRD, which is the hypomethylation 5' to the RAD51C promoter, which leads to alternative isoform expression. And finally, I would like to thank my colleague at Genome Sciences Centre, Andrew and John, more specifically, and the director, Stephen Jones, and also the patients and their families for their trust in our program.